This is a video about meiosis. I'm going to go pretty fast here because I know you can pause it whenever you need to, and I want to just capture all this as quickly as I can. So this is starting on page 14 in your notes packet. In general, meiosis takes a normal cell and reduces the chromosome number in half. So in humans, if a normal human cell has 46 chromosomes, we remember, then each cell at the end of meiosis will have 23 chromosomes. Kind of like this drawing right here. This does not show 46 chromosomes, obviously, just 2 chromosomes, one homologous pair of chromosomes. In fact, let's remind ourselves what that means. One homologous pair. Originally, one of these was inherited from mom and one from dad. I'm going to color them in a little bit. Color one red. Color one green, so we can keep track of them. Oh, red and green, how Christmassy, how nice. But that was what was inherited from mom and dad, and now this individual is going to try to create cells that it could use to pass on the information to its offspring. So we're going to be trying to make sex cells, eggs and sperms, from this. You would be doing this with 46 chromosomes. This little guy is just doing it with 2 chromosomes. Here we go. So the first thing that happens is the chromosomes replicate. So here's now my red one replicated, my green one replicated. I don't want to shade in, that's okay. So this is now one replicated homologous pair. And hopefully you did this successfully in your mitosis video, and so you know what this is talking about. But if you're not sure, there we are, there's a picture of it. Now, in this case, what happens in meiosis is we're going to do 2 divisions after we replicate them. Here's the first division, and then here's the second division. And we call this first division meiosis 1. And then the second division we call meiosis 2. Roman numerals, it's just how they do it. So what happens is first you split up the homologous pairs. So both sister chromatids of the red go one way, and both sister chromatids of the green go the other way and end up in separate cells. And then these cells further split so that the sister chromatids now divide, one into each subsequent cell. How many cells do you get at the end? I count 4. That's very different from mitosis, isn't it? In fact, in mitosis, let's just have a quickie review over here. Here's the original cell for mitosis. So here is, if this was the same sort of thing, we'd have one red and one green chromosome. First thing that happens is they would replicate. And you'd have your green chromosome. Oops, sorry. You'd have your red chromosome. And then at the end of mitosis, you get 2 cells. One of which has a red chromosome and a red chromosome and a green chromosome, and the other one has a red chromosome and a green chromosome, because the point was to make exact copies of the original one. So notice how we've got 2 cells down here with exactly the same number and types of chromosomes as our original cell had. And we did that by duplicating the cells and then splitting them. Over in meiosis, the point is not to get identical to the top one. In fact, what we got was something that was only half of the top one, and we got a little double one of each half. Did that make sense? Yeah. So I got 4 cells. 2 of them have a red chromosome, 2 of them have a green chromosome. And that's half of what you see up here. How does this process occur? Let's turn to the next page. I want to look at the details of this. This is probably the challenging bit. There's a lot to write and draw, but keep up, you will be just fine. And then we'll come back and finish this page a little bit later. So, turning to the next page, you should see meiosis details. You should see a whole bunch of circles and a dividing line across the middle. I'm on the top half of the page right now. Let's see if we can make this all fit. So, where does meiosis occur? Meiosis occurs in the ovaries or the testes, depending on whether you're a female or a male. You'll be making sperms or eggs, obviously. And what happens is we're going to talk about the meiosis details. Top part here, above the dotted line, is meiosis 1. And then down underneath the dotted line is meiosis 2. So let's just hold on to meiosis 2 for a sec. Can you see that there? Yep. So meiosis 1, back at the top, think about, this is the starting cell. In fact, I want one more color pen. So this is my starting cell. We could call it a spermatocyte if we're making sperms. Spermatocyte. So let's actually do that. So this would be in male's testes making spermatocyte. Making sperm, sorry, from a spermatocyte. Let's do 3 pairs of chromosomes. It's a lot to keep track of, but I think it's going to help us understand in the end better what's going on. So you might want 3 different colors of pen. I'm going to do a long pair of blue, a medium pair of red, and kind of a short squiggly pair of green, just so I can keep track. And if you need to go get colored pens, you should pause this and do so. First thing that happens is the chromosomes replicate. So the chromosomes are going to make copies of themselves. They'll end up looking like little X's. So I'm just going to draw sort of quick X's. What are they held together in the center with? If you said centromeres, you were right. It's hard to draw squiggly pairs, but I'm doing my best. So this is all happening in interphase. I'm running out of room over there. Interphase, all right? So in prophase 1, what happens is in meiosis, you get to have prophase, metaphase, anaphase, and telophase, just like in mitosis, but we have to do it in two parts. So we have prophase 1, metaphase 1, anaphase 1, and so forth. And you'll see the others all appear shortly. So in prophase 1, a lot of stuff happens. This is probably the busiest of all of the phases. So over here, I'm going to write prophase 1. And what happens in prophase 1, first of all, exactly the same as mitosis. So by that I mean the nuclear membrane is going to begin to disappear, the chromatin turns into chromosomes, the spindle fibers begin to appear, the centrioles replicate, all that stuff happens here too. So I'm not going to repeat that. You can look back to that. But something that does happen that's unique to meiosis and unique to prophase 1 in meiosis is a process called crossing over. So what I mean by that is homologous pairs, they will form tetrads. I'll draw you a picture of what I mean. They form tetrads, and they do what's called crossing over. They actually swap DNA pieces, so little bits of DNA switch from one chromosome to the other. So DNA pieces get swapped. This sounds strange. The reason this is important, though, is because you'll increase different combinations of genes, and that increases genetic diversity. So there's a benefit. Whoops, I can't spell genetic. There's a benefit to this, genetic diversity. So what I mean by a tetrad is basically what happens is you have a pair of chromosomes, and you have it's homologous pair of chromosomes, and then we've got our sister tetrads. They actually kind of join up together, and they wind their little legs around each other, and they break off bits. They break off the tips and swap them with each other. And when they separate, they've still got all the right numbers of genes and stuff, but they've changed up some of the combinations. Okay, so I'm going to just draw that, kind of sketch that right here. Like here's my blue pair, and I've just got them kind of wound across each other. So that is what I mean by a tetrad. And the red pair do that, and the green pair does that. Oh, this is tough to draw, but I'll do my best. Okay, so that's a big thing that happens in prophase 1. Then they unwind from each other, and they go ahead and do metaphase, anaphase, and telophase. So here's what that looks like. So metaphase 1. This is where homologous pairs will line up across the center of the cell. This is also a little bit different from mitosis. Homologous pairs get together and line up across the middle of the cell. So it's really important actually that they are paired together. So here's my blue pair, here's my red pair, here's my green pair. And the spindle fibers are going to attach, like the ones on this side are only going to attach to this centromere and that centromere, but not to the other pair. Likewise, on the left side, it'll attach to this centromere, that, and that, because what's going to happen is you're going to pull all of these replicated chromosomes one way, and all of these go the other way. But the sister chromatids are not going to be splitting. Some of you in your mitosis projects actually lined your chromosomes up like this, although you then split your sister chromatids correctly. And that actually isn't quite what happens in mitosis, but it does happen in meiosis. All right, next thing that happens is anaphase 1. Anaphase 1, the homologous pairs separate. So when the homologous pairs separate, it looks something like this. Oh, I forgot to write. This is metaphase 1. Here we have anaphase 1. Notice I'm abbreviating because it's just faster. That's fine. So I have centriole. Oh, and my cell got a little wider. Check that out. Let's see. So the blue pair is going to go over this side, that side. Red pair separates like that. Green pair like that. Got to draw some spindle fibers. Okay, so you see how they're splitting, but the sister chromatids have not disassembled yet. So in telophase, this is a little bit odd. Telophase 1 and cytokinesis. You're going to form two cells, but each of which has only half the original homologous pair. Each has only one half of the original homologous pair. And it will look something like right here, down here below the dotted line. So I've got blue here and a blue there, red, red, green, green. And it doesn't quite look like our original cell, does it? So, and it doesn't look like what we should have gotten from mitosis either. What happens is now telophase 1 actually kind of goes right into prophase 2. In some cases, you're going to have kind of a delay. For example, in the ovaries of a female, you might have a delay of months or even years until one egg matures and then it's ready to be fertilized, perhaps. Sometimes it just goes straight from telophase 1 right into prophase 2 without anything in between. So there's not really an interphase. So I'm going to kind of ignore that. I might just write that if there is an interphase, it's just a resting phase. The chromosomes do not get copied. So I'll just make a note of that right there. Okay, so this part's a little bit simpler now. So I'm going to go kind of fast. Prophase 2. Here, it's the same things as happens as mitosis. If the nuclear membrane reformed, it disappears. Again, the spindle fibers reappear, so I'm going to draw some centrioles and get some spindle fibers beginning to grow. Okay. In metaphase 2, the chromosomes line up across the middle of the cell. We have to do it for two cells, of course, because we now have two cells. So in the middle of each cell. So that gives me a blue one here and here, a red one here and here, and there's my green one. What else do I need? Oh, I need centrioles. And this time the spindle fibers will connect from both sides because the sister chromatids are going to be pulled apart. I also have spindle fibers that would be going all the way across, just like we had in mitosis, but I'm not going to draw those just because it gets a little bit crowded. What is the name of this phase? This is metaphase 2. Okay. So the next stage, anaphase 2. We have something has to separate. That's what anaphase does. So in anaphase 2, the sister chromatids will separate. The sister chromatids now separate. So one half of the blue goes this way, one goes that way. Same over here. Same for red. Same for green. And let's get some spindle fibers in there, pulling them along. Remember, the spindle fibers are kind of acting like a little fishing rod, reeling in the chromosomes so that they separate. Okay. So this was anaphase 2. Kind of messy there, but I think we can follow along. You doing okay? Fabulous. So telophase 2. and cytokinesis. In telophase 2 and cytokinesis, basically now that you've got this nuclear membrane reforms, all the normal stuff for telophase happens, but the important thing to note is 4 gametes have formed, and each of them has half the normal number of chromosomes, the original number of chromosomes. Each has only half the original number of chromosomes. So let's sketch that in for telophase. Telophase might look something like, here's my blue ones have separated, my red ones have separated, my green ones have separated. They're turning back into chromatin. The nuclear membrane would be reforming. So here's my nuclear membrane reforming. We would have cytokinesis occurring. So that would look kind of like a split happening. I think some of yours got cut off on the bottom here. I apologize for that. You could draw it in. Kind of hard to draw the splitting in, but I think you know what's going on there. So once these nucleuses have reformed, nuclei have reformed, let's go over here and actually add some flagella, make some sperm out of these. So you get 4 sperm, connect them up at the bottom here, 4 sperm. What's going to go in each, chromosome-wise? If you predicted that you're going to get 1 blue, 1 red, 1 green chromosome in each, you predicted correctly. And that's exactly what happens. So what I want you to do is compare these 4 final cells that you get with the original cell at the very top. And notice that each little sperm is identical to the original cell, except it only has half the chromosomes. But it has the right half. It's got 1 blue, 1 red, 1 green. Not like 2 blues and a red. It has to have 1 blue, 1 red, 1 green. And that's what meiosis does. Back on page 14, you will need to catch up on that at a time other than this video, but you can check the notes, you can check with a friend, you can check with Ms. Robinson, you can check with Ms. O'Shaughnessy. Happy to help you out. I hope this helps you understand meiosis just a smidgen bit better.